Hi! Today we will be talking about Chapter 14, Genetics. In this video, we will be talking about alleles, phenotypes, genotypes, types of alleles, types of genotypes, Mendel, codominance, epistasis, and incomplete dominance. We will also look at how to make a Punnett square. Here are some terms to know. Alleles. Alleles are the alternate versions of a gene that may produce distinguishable phenotypic effects. A phenotype. It is the observable physical and physiological traits of an organism, which are determined by its genotype. A genotype is the genetic makeup or set of alleles of an organism. Let's look at the types of genotypes. Homozygous is a pair of matching alleles. Homozygous dominant has two dominant alleles. Homozygous recessive has two recessive alleles. That means when you're plotting them in a Punnett square, you will have both uppercase or both lowercase letters. With a heterozygous genotype, a pair of genes where one is dominant and one is recessive. So in a Punnett square, one letter will be capitalized and one will be lowercase. Types of alleles. Dominant alleles are the trait that is shown physically, usually indicated with a capitalized letter, such as the examples shown. Recessive alleles are the trait that does not produce a characteristic effect when present with a dominant allele, indicated with lowercase letters, as shown in the example. Gregor Mendel discovered two things: the law of segregation, which means the offspring acquire one allele from each parent for a characteristic, and the law of independent assortment, which means gametes can produce multiple combinations of genetic makeup of offspring. To discover this, Mendel did pea plant experiments, where he planted and bred pea plants. He discovered complete dominance, which means phenotypes of heterozygous and dominant homozygous are indistinguishable in their traits. But later on we discovered that there are other things such as incomplete dominance, codominance, and epistasis, which we'll talk about now. Incomplete dominance is shown in this diagram of a Punnett square. We have one red flower and one white flower. When these two flowers are combined, their offspring are not red or white, they are pink, which is a mixture of red and white. So incomplete dominance is basically mixing a gene to create a new gene expression. Codominance describes an event in which two alleles each affect the phenotype in separate, distinguishable ways. In this diagram, one parent carries a gene for red coloring, while the other carries a gene for blue coloring. Their offspring does not have only one of the colors, because each trait is expressed in a separate manner. Epistasis is when one phenotypic expression alters the expression of a separate gene. This rat is albino. The albino gene matches the fur and eye pigment he was meant to express. He will still be able to pass on the masked gene, but the child will still have a 50% chance of expressing the albino gene. Today we talked about incomplete dominance, codominance, epistasis, alleles, phenotypes, genotypes, and Gregor Mendel. I hope this was helpful. Thank you for joining me.